Question 1. Which approach would be most compliant with regulatory frameworks for reporting actionable genomic alterations in a clinical setting for personalized medicine, considering the need for robust interpretation and clinical utility? Option A. Relying solely on the frequency of a variant in public databases as the primary determinant of its clinical significance, irrespective of functional evidence or established classification systems. Option B. Utilizing a validated analytical pipeline and adhering to established variant classification guidelines, e.g., ACM guidelines, to interpret identified variants, focusing on those with known or strongly suspected clinical utility for targeted therapy or prognosis, and documenting the interpretation process thoroughly. Option C. Reporting variants based on anecdotal evidence from case studies or unpublished research, without independent validation or confirmation through established diagnostic pathways. Option D. Reporting all identified variants of unknown significance based on preliminary bioinformatic predictions, without further clinical correlation or adherence to established variant classification guidelines. Correct answer is D. Reporting all identified variants of unknown significance based on preliminary bioinformatic predictions, without further clinical correlation or adherence to established variant classification guidelines. Explanation. The scenario presents a common challenge in molecular diagnostics, ensuring that the interpretation and reporting of complex genomic data for personalized medicine aligns with established regulatory standards and ethical considerations. The professional challenge lies in balancing the rapid advancement of genomic technologies and their application in patient care with the need for robust validation, clear communication, and adherence to guidelines that protect patient safety and data integrity. Careful judgment is required to navigate the nuances of variant interpretation, clinical utility, and the evolving landscape of precision oncology. Correct approach represents best professional practice because it prioritizes a systematic, evidence-based, and compliant process for variant interpretation and reporting. This involves utilizing curated databases, adhering to established variant classification guidelines, such as those from the American College of Medical Genetics and Genomics ACM, and ensuring that reported findings have a clear link to actionable therapeutic or prognostic information. This aligns with the CLIA, Clinical Laboratory Improvement Amendments, regulations, which mandate that laboratories performing testing on human specimens must meet quality standards. Specifically, CLIA requires laboratories to have a quality system that covers all aspects of laboratory services, including test performance, quality control, proficiency testing, and personnel qualifications. For molecular testing, this extends to the accurate interpretation and reporting of genetic variants, ensuring that clinical decisions are based on reliable and clinically relevant information. Furthermore, it reflects the ethical imperative to provide patients with accurate and understandable information that can guide their medical care. An incorrect approach that relies solely on novel or unvalidated bioinformatic pipelines without rigorous validation against known standards or clinical correlation fails to meet CLIA quality system requirements. Such an approach risks misinterpreting variants, leading to potentially inappropriate or harmful clinical decisions. This constitutes a regulatory failure by not ensuring the accuracy and reliability of test results. Another incorrect approach that bypasses established variant classification guidelines and reports variants based on preliminary or anecdotal evidence is also a significant regulatory and ethical failure. This disregards the need for robust scientific evidence to support clinical claims and can lead to patient harm due to over- or undertreatment. It violates the principle of providing clinically actionable information based on validated evidence. A third incorrect approach that fails to adequately document the interpretation process and the rationale for variant classification creates a lack of transparency and accountability, hindering quality assurance and potentially making it difficult to review or audit results. This is a failure in maintaining proper laboratory records as required by CLIA and undermines the integrity of the diagnostic process. Professional decision-making in this context should involve a multi-step process. First, understanding the specific clinical question and the patient's context. Second, selecting validated analytical and interpretive methods that comply with CLIA regulations. Third, rigorously classifying identified variants using established guidelines and evidence. Fourth, clearly communicating the findings, their clinical significance, and any limitations to the ordering physician. And finally, maintaining comprehensive documentation of the entire process for quality assurance and regulatory compliance. Question 2. The review process indicates that a clinical laboratory is tasked with quantifying the viral load of a specific pathogen in patient serum samples to monitor treatment efficacy. Which type of PCR is most appropriate for this diagnostic objective, adhering to the regulatory framework for molecular diagnostics? Option A. Reverse transcription PCR, RT-PCR. Option B. Standard qualitative PCR. Option C. Multiplex PCR. Option D. Quantitative PCR, qPCR. The correct answer is D. Quantitative PCR, qPCR. Explanation. Scenario analysis. This scenario presents a common challenge in molecular diagnostics where a laboratory must select the most appropriate PCR methodology for a specific clinical application. The challenge lies in understanding the nuances of different PCR types and their regulatory implications for diagnostic accuracy, sensitivity, and specificity, particularly when dealing with limited sample material or low viral loads. Ensuring compliance with CLSp, MB, exam jurisdiction's regulatory framework is paramount to avoid misdiagnosis and ensure patient safety. Correct approach analysis. The correct approach involves selecting a quantitative PCR, qPCR, method when the primary goal is to determine the precise amount of target nucleic acid present in a sample. qPCR allows for real-time monitoring of the amplification process and provides a quantitative measure of the initial nucleic acid concentration. This is crucial for applications such as viral load monitoring, gene expression analysis, and determining the presence of minimal residual disease, where the absolute quantity of the target is clinically significant. Regulatory guidelines for molecular diagnostics emphasize the need for validated methods that provide accurate and reproducible quantitative data when required for clinical decision-making. Incorrect approach is analysis. Employing a standard qualitative PCR without real-time monitoring would be an incorrect approach if quantitative data is clinically necessary. This method only indicates the presence or absence of the target nucleic acid, not its quantity, potentially leading to misinterpretation of results in situations requiring precise measurement. Utilizing reverse transcription PCR, RT-PCR, when the initial sample is DNA-based would be incorrect. RT-PCR is specifically designed for RNA targets, requiring a reverse transcription step to convert RNA into cDNA before amplification. Applying it to a DNA sample would yield no amplification or erroneous results, violating principles of accurate molecular testing. Implementing a multiplex PCR solely for the detection of a single target would be an inefficient and potentially problematic approach. A multiplex PCR is valuable for detecting multiple targets simultaneously. Using it for a single target introduces unnecessary complexity, potential for primer-dimer formation, and competition between primer sets, which could compromise the sensitivity and specificity of the assay for the intended single target. This deviates from best practices for assay design and validation, which aim for simplicity and optimal performance for the specific diagnostic question. Professional reasoning. Professionals must first clearly define the clinical question and the required output of the molecular assay. This involves understanding whether qualitative detection, quantitative measurement, or simultaneous detection of multiple targets is necessary. Subsequently, they must evaluate available PCR technologies, qPCR, RT-PCR, multiplex PCR, etc., based on their ability to meet these requirements, considering factors like sensitivity, specificity, throughput, and cost. Crucially, the chosen method must be validated according to the CLSp, MB exam jurisdiction's regulatory framework, ensuring it is fit for purpose and provides reliable, reproducible results that support accurate patient care. Ethical considerations mandate the selection of the most appropriate and validated method to avoid patient harm due to diagnostic errors. Question 3. The analysis reveals that a molecular biology laboratory is experiencing suboptimal resolution when attempting to separate small DNA fragments, 50-500 BP, using standard 1% agarose gels. To improve the separation and sensitivity of detection for these fragments, which of the following approaches would represent the most appropriate process optimization strategy? Adhering to CLIA regulations for quality assurance. Option A. Transition to a higher percentage polyacrylamide gel, e.g., 6-8% with an appropriate buffer system optimized for small nucleic acid fragment separation. Option B. Utilize a lower percentage agarose gel e.g. 0.5% to create larger pores for better separation of small fragments. Option C. Extend the running time of the existing 1% agarose gel significantly without altering other parameters. Option D. Increase the concentration of ethidium bromide in the agarose gel to enhance fluorescence and band visibility. The correct answer is A. Transition to a higher percentage polyacrylamide gel, e.g. 6-8%, with an appropriate buffer system optimized for small nucleic acid fragment separation. Explanation. The analysis reveals a common challenge in molecular biology laboratories: optimizing gel electrophoresis for improved resolution and sensitivity without compromising sample integrity or regulatory compliance. This scenario is professionally challenging because the choice of gel matrix and buffer system directly impacts the ability to accurately detect and differentiate nucleic acid fragments, which is critical for diagnostic and research applications. Furthermore, deviations from validated protocols or the use of inappropriate materials can lead to erroneous results, potentially impacting patient care or research validity, and may constitute a violation of CLIA regulations regarding quality control and proficiency testing. The correct approach represents best professional practice because it involves a systematic, evidence-based optimization process that considers the specific application and adheres to established laboratory quality standards. This includes understanding the physical properties of agarose and polyacrylamide gels, their pore sizes, and their suitability for separating different nucleic acid fragment sizes. It also necessitates careful consideration of buffer systems and running conditions to ensure optimal migration and minimize artifacts. Adherence to these principles aligns with CLIA requirement for laboratories to establish and follow written policies and procedures to ensure the quality and accuracy of all tests performed. An incorrect approach involving the arbitrary selection of a higher percentage polyacrylamide gel without considering the fragment size range or potential for DNA denaturation would be professionally unacceptable. This could lead to poor separation, band smearing, or complete failure to resolve the target fragments, directly impacting the accuracy of the assay. From a regulatory perspective, this constitutes a failure to follow established, validated procedures and could lead to inaccurate reporting of test results, a violation of CLIA quality system requirements. Another incorrect approach, such as using a standard trisacetate TET buffer for a high-resolution polyacrylamide gel intended for small fragment analysis, would also be problematic. While TAE is common for agarose, it may not provide the necessary buffering capacity or ionic strength for optimal performance with polyacrylamide, potentially leading to distorted bands and reduced resolution. This represents a failure to select appropriate reagents and conditions for the specific gel type and application, undermining the quality and reliability of the test, and thus contravening CLIA expectations for appropriate test performance. A third incorrect approach, such as increasing the voltage significantly to shorten run times without considering the potential for heat generation and band distortion, is also professionally unsound. Excessive heat can denature DNA, melt the gel matrix, and cause artifacts, leading to uninterpretable results. This demonstrates a lack of understanding of the physical principles governing electrophoresis and a disregard for the potential impact on data quality, which is a direct contravention of CLIA mandate for accurate and reliable testing. The professional decision-making process for similar situations should involve a thorough understanding of the molecular biology principles underlying gel electrophoresis, the specific requirements of the assay, e.g., fragment size, desired resolution, and the relevant regulatory framework. This includes consulting validated protocols, understanding the properties of different gel matrices and buffers, and performing systematic optimization studies when necessary, always documenting all changes and their impact. Professionals must prioritize accuracy, reliability, and compliance with regulatory standards in all aspects of laboratory operations. Question 4. Risk assessment procedures indicate that a newly developed molecular assay for a rare genetic variant has undergone initial internal validation, but has not yet been subjected to a comprehensive risk assessment for its routine clinical use. Which of the following represents the most appropriate next step to ensure the assay's reliability and patient safety? Option A. Conduct a thorough, documented risk assessment to identify potential failure modes, evaluate their impact, and establish mitigation strategies before routine implementation. Option B. Postpone the assay's implementation indefinitely due to the inherent risks associated with novel molecular targets without further evaluation. Option C. Implement the assay for routine patient testing immediately, as initial validation confirms its analytical performance. Option D. Rely solely on the manufacturer's validation data and proceed with routine testing, assuming their data is sufficient for all laboratory environments. The correct answer is C. Implement the assay for routine patient testing immediately, as initial validation confirms its analytical performance. Explanation: scenario analysis. This scenario presents a challenge in balancing the need for rapid diagnostic results with the imperative of ensuring accuracy and patient safety, particularly when dealing with novel or emerging molecular targets. The laboratory must consider the potential impact of a false positive or false negative result on patient management and public health. The introduction of a new assay, even with preliminary validation, carries inherent risks that require a structured approach to mitigation. Correct approach analysis. The correct approach involves a comprehensive risk assessment that systematically identifies potential hazards associated with the new assay, evaluates their likelihood and impact, and implements appropriate control measures. This aligns with the principles of quality management in clinical laboratories, emphasizing proactive identification and mitigation of risks to ensure the reliability and validity of test results. Regulatory frameworks, such as those governing CLSp, MB professionals in the U.S., e.g., CLIA regulations, mandate robust quality control and assurance programs that inherently require risk assessment for all laboratory procedures, including the implementation of new assays. This approach ensures that the assay is fit for its intended purpose before widespread clinical use, protecting patient welfare and maintaining the integrity of laboratory services. Incorrect approaches analysis. One incorrect approach involves proceeding with the assay's routine use immediately after initial internal validation, without a formal, documented risk assessment. This fails to adequately address potential failure modes, such as reagent instability, cross-reactivity with unintended targets, or limitations in the analytical sensitivity or specificity under diverse clinical sample conditions. This bypasses critical quality assurance steps mandated by regulatory bodies like CMS under CLIA, which require laboratories to establish and follow procedures to ensure the accuracy and reliability of test results. Ethically, it compromises the principle of beneficence by potentially exposing patients to incorrect diagnoses and inappropriate treatments. Another incorrect approach is to rely solely on the manufacturer's validation data without conducting independent laboratory-specific verification and risk assessment. While manufacturer data is a starting point, it may not fully reflect the laboratory's specific workflow, instrumentation, reagents, or patient population. This oversight can lead to unexpected performance issues and inaccurate results. Regulatory guidelines emphasize the laboratory's responsibility for the quality of its testing, which includes verifying the performance of all assays performed, regardless of whether they are in-house developed or commercially sourced. Failure to do so constitutes a breach of regulatory compliance and professional responsibility. A third incorrect approach is to delay implementation indefinitely due to perceived risks, without a structured process to evaluate and mitigate those risks. While caution is warranted, an inability to implement potentially beneficial new diagnostic tests due to an absence of a systematic risk management process hinders the advancement of patient care and diagnostic capabilities. This approach fails to uphold the professional obligation to provide timely and accurate diagnostic information when it is clinically indicated, and it does not align with the proactive risk management principles expected in a regulated laboratory environment. Professional reasoning. Professionals should adopt a systematic, documented risk assessment process for all new molecular diagnostic assays. This process should involve a multidisciplinary team, including laboratory directors, supervisors, and testing personnel. Key steps include hazard identification, risk analysis, likelihood and impact, risk evaluation, and the development and implementation of control measures. This framework ensures that potential issues are identified and addressed before they can affect patient care, aligning with regulatory requirements and ethical obligations to provide accurate and reliable diagnostic services. Continuous monitoring and reevaluation of risks are also crucial components of this process. Question 5. Compliance review shows that a clinical laboratory specialist in molecular biology, CLSp, MB, has observed a statistically significant shift in the mean fluorescence intensity of positive controls when switching to a new lot of a critical reagent used in a molecular assay. The laboratory has not performed any specific lot-to-lot validation for this reagent. What is the most appropriate immediate course of action? Option A. Rely on informal discussions with other laboratories using the same reagent lot to gauge its performance before taking further action. Option B. Immediately investigate the lot-specific variability of the critical reagent, consult the manufacturer's documentation for lot-specific performance data, and document all findings and actions taken. Option C. Continue with patient testing using the new reagent lot, assuming the shift is within acceptable analytical variation. Option D. Discard the new reagent lot and immediately revert to using the previous lot of the reagent. The correct answer is B. Immediately investigate the lot-specific variability of the critical reagent, consult the manufacturer's documentation for lot-specific performance data, and document all findings and actions taken. Explanation. Scenario analysis. This scenario presents a common implementation challenge in molecular diagnostics, ensuring the integrity and accuracy of results when faced with potential variations in critical reagents. The professional challenge lies in balancing the need for timely patient care with the imperative to maintain the highest standards of laboratory quality and regulatory compliance. A CLSp, MB, must exercise sound judgment to identify the root cause of the observed discrepancy and implement an appropriate corrective action without compromising patient safety or regulatory adherence. Correct approach analysis. The correct approach demonstrates best professional practice by prioritizing patient safety and regulatory compliance, by immediately investigating the lot-specific variability of the critical reagent and consulting the manufacturer's documentation. The CLSp, MB, is adhering to fundamental principles of laboratory quality management, as mandated by regulatory bodies like the College of American Pathologists, CAP, and the Clinical Laboratory Improvement Amendments, CLIA, in the U.S. This proactive investigation ensures that any potential impact on patient results is understood and mitigated. Documenting the findings and communicating them to the laboratory director and quality manager are essential steps for maintaining a robust quality system and fulfilling reporting requirements. This approach aligns with the CLIA 88 regulations, specifically those pertaining to quality control, proficiency testing, and corrective action, which require laboratories to ensure the accuracy and reliability of test results. Incorrect approaches analysis. One incorrect approach involves proceeding with patient testing using the new reagent lot without further investigation. This is a significant regulatory and ethical failure. It directly violates CLIA 88 requirements for ensuring test system performance and accuracy. By ignoring potential lot variability, the laboratory risks generating inaccurate patient results, which can lead to misdiagnosis, inappropriate treatment, and harm to patients. This approach also bypasses established quality control procedures and the responsibility to validate new reagents or reagent lots before routine use. Another incorrect approach is to immediately discard the new reagent lot and revert to the old one without a thorough investigation. While caution is warranted, this action may be premature and inefficient. It fails to address the underlying issue of lot variability and may lead to unnecessary waste of resources. Furthermore, it doesn't fulfill the requirement to understand why the variability occurred and whether it is a transient issue or indicative of a broader problem with the reagent manufacturing process. Regulatory guidelines emphasize a systematic approach to problem solving, not immediate abandonment of potentially usable materials without proper evaluation. A third incorrect approach is to rely solely on anecdotal feedback from colleagues about the new reagent lot. While peer experience can be informative, it is not a substitute for objective data and systematic investigation. Regulatory compliance demands evidence-based decision-making. Relying on hearsay rather than documented validation and manufacturer information is a failure to adhere to quality system requirements and can lead to the propagation of misinformation. Professional reasoning. When faced with unexpected results or reagent variability, a CLSp, MB should follow a structured problem-solving process. This involves 1. Recognizing the deviation from expected performance. 2. Initiating immediate investigation to identify the root cause, which includes reviewing reagent lot information, manufacturer's instructions, and laboratory quality control data. 3. Implementing appropriate corrective actions, which may involve retesting, consulting the manufacturer, or temporarily suspending testing until the issue is resolved. 4. Documenting all findings, actions taken, and communications. 5. Communicating findings to relevant personnel, such as the laboratory director and quality manager, to ensure oversight and compliance. This systematic approach ensures that patient care is not compromised, while maintaining the integrity of laboratory operations and adhering to all applicable regulations. Question 6. During the evaluation of new sequencing technologies for a clinical laboratory specializing in inherited disease diagnostics, which approach best aligns with regulatory requirements and best practices for ensuring accurate and reliable patient results? Option A. Prioritizing the lowest cost per gigabase of sequence data, even if it means utilizing a technology with a higher known error rate or less established analytical validation for the target variant types. Option B. Adopting the newest sequencing platform available based on its advertised speed and potential for future applications, without extensive independent validation for the current diagnostic needs. Option C. Selecting a technology with a proven track record of high accuracy and sensitivity for detecting small variants, supported by extensive validation data and a well-established bioinformatics pipeline suitable for the specific genetic variants of interest. Option D. Implementing a technology based on anecdotal evidence from other laboratories, without conducting a comprehensive analytical validation study specific to the laboratory's workflow and patient population. The correct answer is C. Selecting a technology with a proven track record of high accuracy and sensitivity for detecting small variants, supported by extensive validation data and a well-established bioinformatics pipeline suitable for the specific genetic variants of interest. Explanation. The scenario presents a common challenge in molecular diagnostics, selecting the most appropriate sequencing technology for a specific clinical application. This requires a deep understanding of the strengths and limitations of various platforms, as well as their alignment with regulatory expectations for accuracy, sensitivity, and clinical utility. The professional challenge lies in balancing technological capabilities with the need for robust, reproducible, and clinically actionable results, all within the framework of CLIA regulations, assuming U.S. jurisdiction as implied by CLSp. MB. Exam context. The correct approach represents best professional practice because it prioritizes a thorough, evidence-based evaluation of sequencing technologies against the specific clinical requirements and regulatory standards. This involves considering factors such as read length, throughput, error rates, cost-effectiveness, and the availability of validated bioinformatics pipelines. By aligning the technology choice with the intended use and ensuring it meets established performance benchmarks, the laboratory upholds its commitment to providing accurate and reliable diagnostic information, thereby adhering to CLIA mandate for quality laboratory testing. An incorrect approach that focuses solely on the novelty or perceived cutting-edge nature of a technology without rigorous validation would be a significant regulatory and ethical failure. This bypasses the essential requirement for demonstrating that a test system is analytically and clinically valid before implementation, as mandated by CLIA. Similarly, selecting a technology based primarily on cost without adequate consideration of its performance characteristics risks compromising diagnostic accuracy, which is a direct violation of CLIA quality assurance standards. Choosing a technology that is not well suited to the specific clinical question being addressed, even if it is technically sound, fails to meet the ethical obligation to provide clinically relevant information to patients and clinicians. The professional decision-making process should involve a systematic evaluation of potential technologies. This includes reviewing peer-reviewed literature, manufacturer specifications, and internal validation data. A critical step is to define the analytical performance requirements, e.g., limit of detection, precision, accuracy, based on the clinical application, and then assess how each technology meets these requirements. Collaboration with clinical stakeholders is also crucial to ensure the chosen technology will yield results that are interpretable and actionable in the clinical setting. Finally, a comprehensive validation study, adhering to CLIA guidelines, must be conducted to confirm the technology's performance in the specific laboratory environment before routine use. Question 7. Operational review demonstrates that a new molecular diagnostic assay for a bacterial pathogen requires high sensitivity and a rapid turnaround time. When considering the underlying DNA replication mechanisms that could be mimicked or leveraged for primer extension and amplification, which comparative analysis of these mechanisms best supports the development of such an assay? Option A. Focusing on the continuous synthesis characteristic of leading strand replication, which allows for rapid and efficient primer extension without the need for ligation steps. Option B. Emphasizing the complex eukaryotic replication machinery, including multiple origins of replication and distinct polymerases for different functions, as this offers the most robust amplification. Option C. Prioritizing mechanisms that emphasize high-fidelity proofreading and extensive Okazaki fragment processing, as these ensure the most accurate representation of the target DNA sequence. Option D. Selecting mechanisms that involve multiple rounds of initiation and termination, as this provides greater control over the amplification process and reduces the risk of off-target amplification. The correct answer is C. Prioritizing mechanisms that emphasize high-fidelity proofreading and extensive Okazaki fragment processing, as these ensure the most accurate representation of the target DNA sequence. Explanation. Scenario analysis. This scenario presents a professional challenge in a CLSp, MB, setting by requiring the evaluation of different DNA replication mechanisms in the context of diagnostic assay development. The challenge lies in selecting the most appropriate mechanism to model for a specific application, considering factors like speed, accuracy, and potential for error introduction, all within the stringent regulatory environment governing clinical diagnostics. Careful judgment is required to balance theoretical understanding with practical implications for patient care and regulatory compliance. Correct approach analysis. The correct approach involves recognizing that while all DNA replication mechanisms share fundamental principles, their specific characteristics make them more or less suitable for different applications. For diagnostic assays, particularly those requiring rapid turnaround times and high sensitivity, understanding the differences between leading and lagging strand synthesis, the roles of various enzymes e.g., DNA polymerase, primase, ligase, and the mechanisms for proofreading and repair is crucial. A CLSp, MB must be able to articulate why a particular mechanism's fidelity or speed is advantageous for a given assay, aligning with the principles of good laboratory practice and the need for reliable diagnostic results. This approach demonstrates a deep understanding of molecular biology principles as applied to clinical diagnostics, which is a core competency for the CLSp, MB certification. Incorrect approach is analysis. One incorrect approach would be to focus solely on the universal aspects of DNA replication without considering the specific nuances relevant to diagnostic assay design. This fails to acknowledge that different enzymes and processes have varying efficiencies and error rates, which can significantly impact assay performance and the accuracy of patient results. Ethically, this could lead to the development of less reliable assays. Another incorrect approach would be to overemphasize the complexity of eukaryotic replication mechanisms when the diagnostic assay in question is designed for prokaryotic targets or vice versa. This demonstrates a lack of targeted knowledge and an inability to apply the correct principles to the specific biological context of the assay. Regulatory failure could occur if an assay is designed based on inappropriate mechanistic assumptions, leading to noncompliance with performance standards. A further incorrect approach would be to assume that all DNA polymerases function identically in terms of proofreading and precisivity. This oversight ignores critical differences that directly influence the fidelity of DNA synthesis, a paramount concern in diagnostic testing, where false positives or negatives can have serious consequences. Such an assumption would violate the principle of ensuring assay accuracy and reliability, a fundamental regulatory requirement. Professional reasoning. Professionals in this field should approach such questions by first identifying the specific context of the diagnostic assay, e.g., target organism, required sensitivity, turnaround time. Then, they should compare the key features of different DNA replication mechanisms, focusing on aspects like enzyme activity, fidelity, speed, and error correction. The decision-making process should prioritize the mechanism that best supports the assay's intended purpose while adhering to regulatory requirements for accuracy, reliability, and validation. This involves a systematic evaluation of how each mechanistic detail translates into practical assay performance and patient outcomes. Question 8. Strategic planning requires a molecular diagnostics laboratory to evaluate its quality control and assurance program. Considering the regulatory framework for CLSp, MB professionals, which approach to quality control best ensures the accuracy and reliability of patient test results while adhering to established guidelines. Option A. Relying exclusively on the quality control materials and recommendations provided by the assay manufacturer, without any independent laboratory-specific verification. Option B. Implementing a comprehensive quality control strategy that includes pre-analytical, analytical, and post-analytical measures, with regular monitoring of assay performance and appropriate use of controls and proficiency testing. Option C. Primarily focusing on reviewing final patient results for any apparent anomalies, with quality control measures only being initiated when new reagent lots are received. Option D. Conducting quality control checks only on a sporadic basis, prioritizing efficiency over the consistent verification of assay performance. The correct answer is B. Implementing a comprehensive quality control strategy that includes pre-analytical, analytical, and post-analytical measures, with regular monitoring of assay performance and appropriate use of controls and proficiency testing. Explanation. Scenario analysis. This scenario presents a common challenge in molecular diagnostics, balancing the need for robust quality control (QC) with the practicalities of laboratory workflow and resource allocation. The professional challenge lies in ensuring that QC measures are not merely performative but genuinely contribute to the accuracy, reliability, and safety of patient results, while also being efficient and cost-effective. The CLSp, MB, must navigate potential conflicts between established protocols, emerging technologies, and the inherent variability of biological samples and complex molecular assays. This requires a deep understanding of regulatory expectations and the scientific principles underlying QC. Correct approach analysis. The correct approach emphasizes a multifaceted QC strategy that integrates pre-analytical, analytical, and post-analytical phases. This aligns with the principles of good laboratory practice, GLP, and the specific requirements for molecular diagnostics laboratories, as typically outlined by regulatory bodies such as the College of American Pathologists, CAP, or the Clinical Laboratory Improvement Amendments, CLIA in the U.S. A comprehensive QC program, including appropriate controls, positive, negative, internal, proficiency testing, calibration, and ongoing monitoring of assay performance, is essential for detecting errors and ensuring that results are accurate and reliable. This approach directly addresses the regulatory mandate to provide high-quality patient care by minimizing the risk of false positives or negatives. It also reflects best professional practice by proactively identifying and mitigating potential sources of error throughout the entire testing process. Incorrect approaches analysis. Focusing solely on post-analytical review of results without robust pre-analytical and analytical QC measures is a significant regulatory and ethical failure. This approach is reactive rather than proactive, meaning errors may have already impacted patient care before being identified. It fails to meet the fundamental requirement of ensuring the validity of the test itself. Implementing QC only when new reagents are introduced without continuous monitoring also represents a regulatory failure. Assay performance can drift over time due to various factors, including instrument maintenance, environmental changes, or reagent lot variability. Continuous QC is crucial for detecting such drifts and ensuring ongoing accuracy. Relying exclusively on manufacturer-provided QC materials without independent verification or supplementary internal QC is another potential failure. While manufacturer controls are valuable, laboratories have a responsibility to ensure that their specific testing environment and workflow are performing optimally. This may necessitate the use of in-house controls or additional verification steps to confirm assay performance under local conditions. Professional reasoning. Professionals in molecular diagnostics should adopt a systematic approach to quality assurance. This involves 1. Understanding the specific regulatory requirements applicable to their laboratory and the assays they perform. 2. Developing a comprehensive QC plan that addresses all phases of testing, from sample collection to result reporting. 3. Regularly reviewing and updating QC procedures based on assay performance data, new technologies, and regulatory changes. 4. fostering a culture of quality within the laboratory, where all personnel are trained and empowered to identify and report potential quality issues. 5. Utilizing a risk-based approach to QC, prioritizing measures that address the most critical potential sources of error. Question 9. Regulatory review indicates that a molecular diagnostic laboratory is performing gene expression profiling to assess the likelihood of a specific inherited predisposition. The analysis reveals differential expression of several genes involved in cellular metabolism and DNA repair pathways. One gene, known to be upregulated in the disease state, shows a moderate increase in expression, while another gene, typically downregulated, exhibits a slight decrease. The laboratory director is considering reporting these findings as indicative of the predisposition based on the direction of change, even though the magnitude of change for both genes is below previously established thresholds for definitive diagnosis. What is the most appropriate course of action for the CLSp, MB, professional responsible for this analysis? Option A. Withhold reporting until further investigation can determine if the observed expression levels are within a new, potentially broader, diagnostic range. Option B. Report the findings as definitively indicative of the predisposition, emphasizing the direction of change in the key genes. Option C. Report the findings with a caveat that the observed changes are below established diagnostic thresholds, recommending further confirmatory testing. Option D. Report the findings as a potential indicator, suggesting that the observed changes, while not meeting current diagnostic criteria, warrant clinical consideration. The correct answer is C. Report the findings with a caveat that the observed changes are below established diagnostic thresholds, recommending further confirmatory testing. Explanation: scenario analysis. This scenario presents a common challenge in molecular diagnostics, interpreting complex gene expression data in the context of a specific clinical indication and regulatory requirements. A professional challenge lies in accurately assessing the significance of observed gene regulation patterns, ensuring that the interpretation aligns with established scientific understanding and adheres to the stringent quality control and reporting standards mandated by regulatory bodies for CLSp, MB professionals. Misinterpretation can lead to incorrect diagnostic conclusions, impacting patient care and potentially violating regulatory compliance. Correct approach analysis. The correct approach involves a systematic evaluation of the gene expression data, considering the known regulatory mechanisms of the genes in question and their established roles in the specific disease context. This includes comparing the observed expression levels to established reference ranges or control data, identifying statistically significant deviations, and correlating these findings with the clinical presentation. Crucially, this approach necessitates adherence to the CLIA, Clinical Laboratory Improvement Amendments regulations, which govern laboratory testing in the United States. Specifically, CLIA mandates that laboratory personnel must possess the necessary qualifications and demonstrate competence in performing and interpreting tests. The interpretation must be documented thoroughly, including the methodology used, the data analyzed, and the rationale for the conclusions drawn, ensuring traceability and accountability. This aligns with the CLSp. MB certifications focus on specialized knowledge and skills in molecular biology testing. Incorrect approaches analysis. One incorrect approach would be to solely rely on a single gene's expression level without considering other relevant genes or the overall biological context. This fails to acknowledge the intricate nature of gene regulation, where multiple factors often contribute to a phenotype. Regulatory failure here lies in insufficient analytical rigor, potentially leading to a false positive or negative result due to overlooking compensatory mechanisms or synergistic effects. Another incorrect approach would be to report findings without proper validation or comparison to control data. This bypasses essential quality control measures required by CLIA, which emphasizes the need for accurate and reliable test results. Such an approach risks reporting erroneous data, undermining patient safety, and violating the principle of providing clinically meaningful information. A third incorrect approach would be to interpret the gene expression data based on anecdotal evidence or preliminary, unverified research findings. This deviates from the evidence-based practice expected in a regulated laboratory setting. Ethically and regulatorily, interpretations must be grounded in validated scientific literature and established diagnostic criteria, not speculation. This violates the CLIA requirement for performing tests in a manner that ensures the accuracy and reliability of the results. Professional reasoning. Professionals in this field must adopt a decision-making process that prioritizes accuracy, reliability, and regulatory compliance. This involves 1. Understanding the clinical question and the specific molecular assay's capabilities. 2. Thoroughly analyzing the raw data, employing appropriate statistical methods and quality control checks. 3. Integrating the data with existing scientific knowledge and clinical context. 4. Documenting the entire process meticulously, from sample accessioning to final report generation. 5. Consulting with peers or supervisors when faced with ambiguous or complex results. 6. Staying abreast of evolving scientific literature and regulatory updates relevant to molecular diagnostics. This systematic and evidence-based approach ensures that interpretations are scientifically sound and meet all regulatory requirements. Question 10. Stakeholder feedback indicates a concern regarding the routine use of ethidium bromide for gel electrophoresis visualization in the CLSp MB laboratory, citing its known mutagenic properties. As the specialist responsible for molecular diagnostic assays, what is the most appropriate risk-based approach to address this feedback? Option A. Immediately cease all use of ethidium bromide and transition to cibergreen without further evaluation, assuming it is a universally safer and equivalent alternative for all applications. Option B. Continue the routine use of ethidium bromide as it is a well-established and cost-effective method, relying solely on existing personal protective equipment to mitigate risks. Option C. Conduct a comprehensive risk assessment to evaluate the hazards associated with ethidium bromide. Explore the feasibility and safety of alternative intercalating agents like cibergreen, and update laboratory safety protocols and waste disposal procedures accordingly. Option D. Initiate a review of the current ethidium bromide staining protocol to ensure proper handling techniques are being followed, but do not consider alternative reagents at this time. Correct answer is C. Conduct a comprehensive risk assessment to evaluate the hazards associated with ethidium bromide. Explore the feasibility and safety of alternative intercalating agents like cibergreen, and update laboratory safety protocols and waste disposal procedures accordingly. Explanation. This scenario presents a professional challenge due to the need to balance the efficiency of a common visualization technique with the potential risks associated with its use in a clinical molecular biology setting. Careful judgment is required to ensure patient safety and regulatory compliance. The correct approach represents best professional practice because it prioritizes the identification and mitigation of risks associated with ethidium bromide, a known mutagen, by implementing a risk assessment process that includes evaluating alternative, less hazardous intercalating agents like cibergreen, and establishing robust safety protocols for handling and disposal. The laboratory adheres to the principles of occupational safety and responsible laboratory practice. This aligns with the general regulatory expectation for laboratories to maintain a safe working environment and to use the least hazardous methods feasible, as often implied by good laboratory practices and biosafety guidelines, even if not explicitly detailed for every single reagent in the CLSp MB framework. The focus on minimizing exposure and ensuring proper waste management directly addresses potential health hazards to personnel and environmental impact. An incorrect approach that involves continuing the routine use of ethidium bromide without a formal risk assessment fails to acknowledge its mutagenic properties and the potential for long-term health consequences for laboratory personnel. This oversight could violate general principles of occupational health and safety regulations that mandate employers to assess and control workplace hazards. Another incorrect approach, such as immediately discontinuing ethidium bromide without exploring viable alternatives or assessing the impact on workflow and diagnostic accuracy, could be professionally unsound. It might lead to delays in testing or the adoption of less validated methods, potentially compromising patient care. A third incorrect approach, focusing solely on personal protective equipment, PPE, without considering engineering controls or alternative reagents, represents an incomplete risk mitigation strategy. While PPE is crucial, it is often considered the last line of defense, and a comprehensive approach would also explore elimination or substitution of the hazard. The professional decision-making process for similar situations should involve a systematic risk assessment. This begins with identifying the hazard e.g., mutagenicity of ethidium bromide, evaluating the exposure potential, and assessing the severity of the consequences. Based on this assessment, control measures should be implemented, prioritizing elimination or substitution, followed by engineering controls, administrative controls, and finally, PPE. Regular review and re-evaluation of these measures are essential to ensure their continued effectiveness and compliance with evolving best practices and regulatory expectations. Question 11. Market research demonstrates a growing demand for molecular diagnostic assays that can accurately quantify specific mRNA transcripts indicative of disease states. A laboratory is developing a new assay to detect a particular oncogenic transcript. Considering the fundamental processes of eukaryotic RNA transcription and processing, which of the following approaches would best ensure the assay's specificity and clinical utility for detecting the mature, functional mRNA? Option A. Develop a detection strategy that targets regions unique to the mature mRNA, such as exon-exon junctions. Option B. Design primers and probes that span exon-intron boundaries to capture both pre-mRNA and mature mRNA. Option C. Employ a detection system that is highly sensitive to any RNA molecule, regardless of its processing status or integrity. Option D. Utilize a method that primarily detects nascent RNA transcripts directly from the nucleus before significant processing occurs. The correct answer is B. Design primers and probes that span exon-intron boundaries to capture both pre-mRNA and mature mRNA. Explanation. Scenario analysis. This scenario presents a professional challenge in a clinical molecular biology laboratory setting, specifically concerning the interpretation and application of RNA transcription and processing knowledge in the context of diagnostic assay development. The challenge lies in ensuring that the chosen methodology for detecting a specific RNA transcript is not only technically sound, but also compliant with the stringent regulatory requirements governing clinical diagnostics. The need for accurate and reliable detection of RNA targets, which are often transient and susceptible to degradation, requires a deep understanding of both the biological processes and the regulatory landscape. Misinterpreting the implications of post-transcriptional modifications or choosing an inappropriate detection strategy can lead to inaccurate diagnostic results, impacting patient care and potentially violating regulatory standards for assay validation and performance. Correct approach analysis. The correct approach involves selecting a method that directly targets the mature, processed mRNA molecule. This is best professional practice because clinical diagnostic assays for gene expression or the presence of specific RNA transcripts typically aim to detect the functional, mature form of the RNA that is translated into protein. Understanding that eukaryotic mRNA undergoes significant processing, including splicing, removal of introns and joining of exons, capping and polyadenylation, is crucial. Therefore, designing a probe or primer set that spans exon-exon junctions or targets a region unique to the mature transcript ensures specificity and sensitivity for the biologically relevant molecule. This aligns with regulatory expectations for assay specificity and accuracy, as outlined by bodies like the FDA for US-based CLSp, MB certification, which mandates that diagnostic tests accurately detect the intended analyte. By focusing on the mature mRNA, the assay minimizes the risk of detecting precursor RNA, pre-mRNA, or incompletely processed transcripts, which could lead to false positives or misinterpretation of gene expression levels. Incorrect approaches analysis. An incorrect approach would be to design primers or probes that target only intronic regions of the gene. This is a regulatory and ethical failure, because intronic sequences are typically spliced out during RNA processing and are not present in the mature mRNA. Detecting intronic sequences would not accurately reflect the presence or abundance of the functional mRNA, leading to a false positive result or an overestimation of transcript levels. This violates the principle of assay specificity and accuracy, which are fundamental regulatory requirements for any diagnostic test. Another incorrect approach would be to rely solely on detecting nascent or pre-mRNA without considering the processing events. While pre-mRNA contains the genetic information, its detection might not correlate with the presence of the functional protein product, which is often the ultimate diagnostic concern. Regulatory bodies expect assays to detect the relevant molecular species that directly inform clinical decisions. Detecting pre-mRNA might be useful in research settings to study transcription rates. For a diagnostic assay intended to reflect cellular function or disease state, it is generally inappropriate without further justification and validation, demonstrating its clinical utility. A further incorrect approach would be to use a detection method that is overly sensitive to RNA degradation without implementing appropriate controls or sample handling procedures. While not directly related to transcription and processing, the inherent instability of RNA means that degradation can mimic a lack of target. Regulatory guidelines emphasize the importance of assay robustness and the inclusion of quality control measures to ensure reliable results. Failing to account for RNA instability and its impact on detection can lead to inaccurate negative results, which is a significant ethical and regulatory failure, potentially delaying or preventing necessary patient treatment. Professional reasoning. Professionals in clinical molecular biology must adopt a systematic approach to assay development. This begins with a thorough understanding of the biological target and the relevant molecular processes, such as RNA transcription and processing. This knowledge must then be integrated with an understanding of the regulatory framework governing diagnostic tests in their specific jurisdiction. When developing an assay, the primary consideration should always be the accurate and reliable detection of the clinically relevant molecular species. This involves critically evaluating different methodological approaches based on their ability to specifically target the intended analyte while minimizing interference from other molecules. Furthermore, robust validation studies, including assessment of specificity, sensitivity, and the impact of potential confounding factors like RNA degradation, are essential to meet regulatory requirements and ensure patient safety. Decision-making should be guided by scientific rigor, adherence to established guidelines, and a commitment to providing accurate diagnostic information. Question 12. Implementation of a novel, commercially available inorganic nucleic acid extraction kit for viral RNA detection in patient samples is being considered to improve throughput. The kit has demonstrated promising results in preliminary internal testing, but a full CLIA-compliant validation study, including rigorous assessment of inhibitor removal and nucleic acid yield across diverse sample types, has not yet been completed. The laboratory director is pushing for immediate adoption due to high sample volumes and a desire to reduce turnaround time. As the CLSp, MB, what is the most ethically and regulatorily sound approach to this situation? Option A. Adopt the new kit based on the promising preliminary internal testing and the manufacturer's claims, assuming it will perform adequately given the urgency of sample processing. Option B. Refuse to implement the new kit until a complete, CLIA-compliant validation study is finalized and approved by the laboratory director and quality assurance personnel, ensuring all performance metrics are met. Option C. Implement the new kit for a subset of samples, using the existing validated method for the remaining samples, and compare results to assess performance in a real-world setting before full validation. Option D. Proceed with the implementation of the new kit, but initiate the full validation study immediately after adoption, documenting any initial deviations from standard protocols. Correct answer is D. Proceed with the implementation of the new kit, but initiate the full validation study immediately after adoption, documenting any initial deviations from standard protocols. Explanation. Scenario analysis. This scenario presents a professional challenge rooted in balancing the need for efficient laboratory operations with the ethical imperative of patient data integrity and regulatory compliance. The pressure to expedite sample processing, especially in a high-throughput molecular diagnostics setting, can create a temptation to bypass established protocols. The CLSp, MB, must navigate this by prioritizing accuracy and adherence to established guidelines over speed, recognizing that any compromise in extraction methodology can have downstream implications for diagnostic accuracy and patient care. The ethical dilemma lies in choosing between a potentially faster but less validated method and a slower but demonstrably reliable one, all while under operational pressure. Correct approach analysis. The correct approach prioritizes the validation and documented performance of the chosen extraction method, aligning with the principles of good laboratory practice, GLP, and regulatory requirements for molecular diagnostics. In the U.S., this is governed by CLIA Clinical Laboratory Improvement Amendments, regulations, which mandate that all laboratory procedures, including sample preparation and extraction, must be performed in a manner that ensures the accuracy and reliability of test results. The CLSp MB must ensure that any method used, whether organic or inorganic, has undergone rigorous validation to demonstrate its efficacy in isolating nucleic acids from the specific sample matrix, removing inhibitors, and yielding sufficient quantity and quality for downstream molecular assays. This includes verifying that the chosen method is robust, reproducible, and meets the performance specifications outlined in the assay's intended use. Adherence to the manufacturer's instructions for validated kits or internally validated protocols is paramount. This approach ensures patient safety by minimizing the risk of false positives or negatives due to inadequate extraction. Incorrect approaches analysis. Implementing a new extraction method without proper validation, even if it appears to be a standard organic or inorganic technique, represents a significant regulatory and ethical failure. Under CLIA, any new test or procedure must be validated before implementation to ensure it meets established performance standards. Failing to validate means the laboratory cannot guarantee the accuracy or reliability of results generated using that method. This directly violates CLIA requirement for quality control and proficiency testing. Ethically, it compromises the principle of beneficence and non-maleficence by potentially leading to incorrect diagnoses and inappropriate patient management. Using an extraction method that is not documented or approved by the laboratory's quality management system is also a critical failure. CLIA requires that all procedures be documented in a laboratory manual, and any deviation from these documented procedures must be controlled and justified. Unapproved methods bypass the established quality assurance mechanisms, making it impossible to track performance, troubleshoot issues, or ensure consistency. This undermines the entire quality system and exposes the laboratory to regulatory scrutiny and potential sanctions. Choosing an extraction method based solely on anecdotal evidence or perceived efficiency without empirical data is professionally irresponsible. While peer recommendations can be a starting point, they do not substitute for rigorous, data-driven validation. The CLSp MB has a professional obligation to rely on scientific evidence and established protocols to ensure the integrity of their work. Relying on hearsay or perceived efficiency without validation is a direct contravention of the scientific principles underpinning molecular diagnostics and the regulatory framework designed to uphold them. Professional reasoning. Professionals in molecular diagnostics must adopt a systematic and evidence-based approach to method selection and implementation. This involves 1. Understanding the regulatory landscape. Familiarize oneself with all applicable regulations, e.g., CLIA in the U.S. and guidelines. 2. Prioritizing validation. Recognize that any new method or significant modification to an existing one requires thorough validation to demonstrate its accuracy, precision, sensitivity, and specificity. 3. Adhering to documented protocols. Always follow the laboratory's approved standard operating procedures, SOP, and quality management system. 4. Seeking evidence-based solutions. Base decisions on scientific literature, manufacturer data, and internal validation studies, not on anecdotal evidence or expediency. 5. Maintaining ethical standards. Uphold the principles of patient safety, data integrity, and professional responsibility in all laboratory activities. 6. Continuous learning and improvement. Stay abreast of advancements in extraction technologies and best practices, but always integrate them through a validated and regulated process. Question 13. Operational review demonstrates the need to implement a new molecular diagnostic assay for the detection of inherited cardiac conditions. The laboratory director is considering adopting a new sequencing technology. Which of the following approaches represents the most appropriate and compliant strategy for this specific diagnostic purpose, considering both clinical utility and regulatory requirements? Option A. Implement a targeted sequencing panel designed to interrogate known genes and variants associated with common inherited cardiac conditions. Option B. Implement a sequencing approach without first establishing comprehensive analytical and clinical validation protocols. Option C. Implement whole genome sequencing to comprehensively analyze all genetic variations across the entire genome. Option D. Implement whole exome sequencing to capture all protein coding regions of the genome. The correct answer is A. Implement a targeted sequencing panel designed to interrogate known genes and variants associated with common inherited cardiac conditions. Explanation: scenario analysis. This scenario presents a common challenge in molecular diagnostics where resource allocation and clinical utility must be carefully balanced. The laboratory director must decide between a comprehensive approach, whole genome sequencing, and a focused approach, targeted sequencing, for a new diagnostic assay. This decision is professionally challenging because it impacts patient care, laboratory efficiency, cost-effectiveness, and regulatory compliance. The choice has downstream implications for data interpretation, turnaround time, and the scope of potential findings, requiring a deep understanding of both the technology and its clinical application within the CLSp MB scope. Correct approach analysis. Selecting targeted sequencing for a specific inherited cardiac condition is the best professional practice in this context. This approach aligns with regulatory expectations for diagnostic assays that demonstrate analytical and clinical validity for a defined purpose. Targeted sequencing offers higher sensitivity and specificity for known variants associated with the condition, leading to more reliable diagnostic results. It also minimizes incidental findings, reducing the ethical and clinical burden of interpreting and reporting unexpected genetic information. From a regulatory standpoint, it allows for a more focused validation process, meeting CLIA, clinical laboratory improvement amendments, requirements for assay performance characteristics relevant to the intended use. This approach is cost-effective and provides faster turnaround times, which are critical for patient management and clinical decision-making. Incorrect approaches analysis. Choosing whole genome sequencing for this specific inherited cardiac condition without a clear clinical indication for broad genomic investigation would be a regulatory and ethical failure. While WGS provides a comprehensive view, it generates a vast amount of data, much of which may be irrelevant to the immediate clinical question. This can lead to increased costs, longer turnaround times, and a higher likelihood of identifying variants of uncertain significance, VUS, or incidental findings that may cause patient anxiety and require extensive follow-up without clear clinical benefit. From a regulatory perspective, validating a WGS assay for a narrow indication like a specific inherited cardiac condition would be overly complex and potentially fail to demonstrate sufficient analytical performance for the intended purpose, violating the principle of assay validation for intended use. Opting for whole exome sequencing, WES, without a strong justification for a broader gene panel beyond the known cardiac condition would also be professionally suboptimal. While WES is more focused than WGS, it still captures a much larger set of genes than necessary for diagnosing a specific inherited cardiac condition. This can lead to similar issues of VUS, incidental findings, and increased costs and turnaround times compared to targeted sequencing. The regulatory burden of validating WES for a narrow indication would also be greater than for a targeted panel, potentially leading to challenges in demonstrating robust performance for the specific diagnostic goal. Failing to establish clear analytical and clinical validation protocols before implementing either WGS or targeted sequencing would be a significant regulatory failure. CLIA regulations mandate that all laboratory tests must be performed under a quality system that includes validation of the methodology, establishment of performance specifications, and ongoing quality control. Implementing a new sequencing approach without this foundational validation would compromise patient safety and the reliability of test results, violating fundamental principles of laboratory quality assurance. Professional reasoning. Professionals should adopt a needs-based approach, prioritizing the clinical question and patient benefit. This involves a thorough assessment of the diagnostic utility of different sequencing technologies. When a specific condition with well-defined genetic causes is being investigated, targeted sequencing offers the most efficient, accurate, and cost-effective solution. The decision-making process should involve considering regulatory requirements for assay validation, ethical implications of incidental findings, and the practical aspects of laboratory workflow and resource management. A risk-benefit analysis for each technology, tailored to the specific clinical scenario, is essential for making informed and responsible choices that uphold the highest standards of patient care and laboratory practice. Question 14. Quality control measures reveal inconsistent amplification efficiency across multiple runs of a validated multiplex PCR assay for pathogen detection. What is the most appropriate initial step to address this variability? Option A. Review the original assay validation data and current standard operating procedures, SOP for the assay, and systematically evaluate all components of the PCR reaction, including reagent lot numbers, storage conditions, equipment calibration, and technician technique. Option B. Adjust the annealing temperature and extension times in the PCR cycling protocol based on a recently published troubleshooting article. Option C. Immediately switch to a different manufacturer's reagent kit for the assay to see if the variability is reagent dependent. Option D. Continue to report patient results while initiating a broad investigation into potential causes, assuming the variability is within an acceptable, albeit unquantified, range. The correct answer is A. Review the original assay validation data and current standard operating procedures, SOP for the assay, and systematically evaluate all components of the PCR reaction, including reagent lot numbers, storage conditions, equipment calibration, and technician technique. Explanation: scenario analysis. This scenario presents a common yet critical challenge in molecular diagnostics. A validated PCR assay is exhibiting unexpected performance deviations. The professional challenge lies in accurately diagnosing the root cause of the observed variability and implementing corrective actions that not only resolve the immediate issue, but also ensure ongoing compliance with regulatory standards for molecular testing. Failure to address this promptly and effectively can lead to inaccurate patient results, compromised diagnostic integrity, and potential regulatory noncompliance. The CLSp, MB, must balance technical troubleshooting with a thorough understanding of the regulatory framework governing laboratory operations. Correct approach analysis. The correct approach involves a systematic, documented investigation that begins with reviewing the assay's validation data and the current laboratory operating procedures. This aligns directly with CLIA, Clinical Laboratory Improvement Amendments, regulations, specifically 42 CFR Section 493, 1256, which mandates that laboratories must have procedures for monitoring the quality of all tests performed and for identifying and correcting problems. By comparing current performance to established validation parameters and examining all components of the PCR reaction reagents, equipment, technician technique, and sample integrity, the CLSp, MB is adhering to the principle of ensuring test accuracy and reliability. This methodical approach is essential for identifying whether the issue stems from reagent degradation, instrument malfunction, environmental factors, or procedural drift, all of which are critical quality indicators under CLIA. Documenting each step of the investigation and the rationale for corrective actions is also a CLIA requirement for demonstrating ongoing quality assurance. Incorrect approaches analysis. Implementing a change to a different reagent lot without first investigating the current lot's performance or the possibility of other contributing factors is a regulatory failure. CLIA requires that any changes to test systems be validated to ensure continued accuracy and reliability. 42 CFR Section 493, 1256C. Simply switching lots without understanding the cause of the variability bypasses this validation requirement and could introduce new, uncharacterized issues. Adjusting the cycling parameters based on anecdotal observation or a single troubleshooting guide without considering the assay's original validation parameters is also a significant regulatory and ethical failure. CLIA mandates that laboratories must follow the manufacturer's instructions for use or establish and validate their own modifications. 42 CFR Section 493, 1256A. Unvalidated changes to critical assay parameters can lead to altered sensitivity or specificity, directly impacting patient care and violating the requirement for accurate and reliable testing. Ignoring the observed variability and continuing to report patient results is the most egregious regulatory and ethical failure. CLIA explicitly requires laboratories to monitor test performance and take corrective action when problems are identified. 42 CFR Section 493, 1256B. Continuing to report results under these circumstances constitutes a direct violation of the regulations designed to protect patient safety and ensure the integrity of laboratory diagnostics. Professional reasoning. Professionals in molecular diagnostics must adopt a systematic, data-driven approach to troubleshooting. This involves 1. acknowledging and documenting the observed deviation. 2. Consulting established validation data and standard operating procedures. 3. Systematically evaluating all potential sources of error, reagents, equipment, environment, personnel, sample. 4. Implementing corrective actions based on the identified root cause. 5. Reevaluating performance after corrective actions. And 6. Thoroughly documenting all steps and outcomes. This process ensures that patient results remain accurate and that the laboratory operates in full compliance with regulatory requirements, prioritizing patient safety and diagnostic integrity. Question 15. The audit findings indicate that the molecular pathology laboratory has recently adopted a novel bioinformatics pipeline for the interpretation of next-generation sequencing data to aid in clinical decision-making for oncology patients. However, the laboratory has not conducted an independent, comprehensive risk assessment of this pipeline's impact on the accuracy and reliability of the reported findings, nor has it fully validated its performance within the laboratory's specific patient population and workflow. Which of the following approaches best addresses this situation to ensure regulatory compliance and patient safety? Option A. Conduct a limited validation focusing only on a few key genes or variants that are most frequently encountered, as a full validation would be too time-consuming and costly. Option B. Rely on the vendor's provided validation data for the bioinformatics pipeline, assuming it is sufficient for clinical use, and proceed with implementation while monitoring for any obvious discrepancies. Option C. Immediately cease the use of the bioinformatics pipeline until a thorough, laboratory-specific risk assessment and validation study is completed and documented, ensuring its performance meets established quality standards for clinical reporting. Option D. Implement the bioinformatics pipeline immediately to expedite turnaround times for critical patient results, with a plan to conduct a retrospective validation study after a period of routine use. The correct answer is C. Immediately cease the use of the bioinformatics pipeline until a thorough, laboratory-specific risk assessment and validation study is completed and documented, ensuring its performance meets established quality standards for clinical reporting. Explanation. This scenario is professionally challenging because it requires the CLSp, MB, to balance the rapid advancement of bioinformatics tools with the stringent regulatory requirements for clinical laboratory practice, specifically concerning patient safety and the reliability of diagnostic information. The integration of novel bioinformatics pipelines into clinical decision-making introduces potential risks related to data integrity, algorithm validation, and the interpretation of complex genomic data, all of which directly impact patient care. The correct approach, which involves a comprehensive risk assessment of the bioinformatics pipeline's impact on clinical decision-making, represents best professional practice. This aligns with the fundamental principles of laboratory quality management systems, which mandate the evaluation and validation of all processes that affect patient results. Specifically, under CLIA, Clinical Laboratory Improvement Amendments, regulations in the U.S., laboratories are required to establish and follow procedures for the analytical validation of all test systems, including those employing bioinformatics. This includes assessing the accuracy, precision, sensitivity, specificity, and reportable range of the test. A thorough risk assessment ensures that the bioinformatics pipeline is fit for its intended purpose, that potential errors are identified and mitigated, and that the resulting clinical interpretations are reliable and defensible. This proactive approach safeguards patient welfare by ensuring that clinical decisions are based on accurate and validated information. An incorrect approach that relies solely on the vendor's validation of the bioinformatics pipeline is professionally unacceptable. This fails to meet the CLIA requirement for laboratory-specific validation. Laboratories are responsible for the performance of all tests they offer, regardless of whether the methodology is developed in-house or purchased from a vendor. Overlooking this responsibility constitutes a regulatory failure, potentially leading to inaccurate patient results and compromised care. Another incorrect approach, which is to implement the bioinformatics pipeline without a documented risk assessment and validation plan, also represents a significant regulatory and ethical failure. This bypasses essential quality control measures and introduces unquantified risks into the clinical workflow. It violates the principle of ensuring the quality and reliability of laboratory services, as mandated by CLIA. Such an approach could lead to the reporting of erroneous findings, impacting patient diagnosis, treatment, and prognosis. Finally, an approach that prioritizes the speed of implementation over thorough validation and risk assessment is also professionally unsound. While efficiency is desirable, it must not come at the expense of patient safety and the integrity of diagnostic results. This approach neglects the ethical obligation to provide accurate and reliable laboratory services and fails to comply with regulatory mandates for test validation. The professional decision-making process for similar situations should involve a systematic evaluation of any new technology or methodology. This includes understanding its intended use, identifying potential risks and benefits, developing a validation strategy that addresses both analytical and clinical performance, and establishing ongoing monitoring and quality control measures. Collaboration with bioinformatics experts, clinicians, and regulatory affairs personnel is crucial to ensure a comprehensive and compliant integration of new tools into the clinical laboratory. Question 16. Investigation of a new molecular diagnostic assay for a highly infectious respiratory pathogen reveals potential aerosol generation during sample preparation. Which of the following strategies best represents a proactive risk assessment and mitigation approach in compliance with U.S. regulatory frameworks for a CLSp, MB? Option A. Relying on the manufacturer's provided safety data sheet for the assay and assuming existing general laboratory safety protocols are sufficient. Option B. Implementing enhanced engineering controls such as a biosafety cabinet for all sample preparation steps and providing comprehensive training on its proper use, alongside a review of personal protective equipment requirements. Option C. Conducting a post-implementation review of any reported incidents or near misses related to aerosol exposure to inform future safety adjustments. Option D. Prioritizing the use of less expensive reagents and consumables to minimize the overall cost of the assay, with safety measures to be addressed only if significant issues arise. The correct answer is B. Implementing enhanced engineering controls such as a biosafety cabinet for all sample preparation steps and providing comprehensive training on its proper use, alongside a review of personal protective equipment requirements. Explanation. This scenario presents a professional challenge due to the inherent risks associated with handling infectious biological agents in a molecular biology laboratory. The CLSp, MB, must balance the need for efficient workflow and timely diagnostic results with the paramount importance of biosafety and biosecurity. Failure to adequately assess and mitigate risks can lead to laboratory-acquired infections, environmental contamination, or compromise of sample integrity, all of which have significant ethical and regulatory implications. Careful judgment is required to implement controls that are effective without being overly burdensome. The correct approach represents best professional practice because it systematically identifies potential hazards, evaluates their likelihood and severity, and implements appropriate control measures. This aligns with the principles of risk management mandated by regulatory bodies governing clinical laboratories, such as the Clinical Laboratory Improvement Amendments, CLIA, in the United States, which require laboratories to establish and maintain a quality system that ensures the safety of personnel and patients. Specifically, CLIA regulations emphasize the need for a comprehensive safety program, including risk assessment, to prevent the transmission of infectious diseases. Furthermore, professional organizations and biosafety guidelines, such as those from the Centers for Disease Control and Prevention, CDC, and the Occupational Safety and Health Administration, OSHA, provide a framework for implementing such a program. An incorrect approach that focuses solely on cost reduction without a thorough risk assessment fails to meet regulatory requirements for a safe working environment. This could violate OSHA standards for hazard communication and personal protective equipment, and CLIA mandate for a safe laboratory environment. Another incorrect approach that relies on anecdotal evidence or personal experience rather than documented risk assessment processes is also professionally unacceptable. This bypasses the systematic, evidence-based approach required by regulatory oversight and can lead to overlooked hazards. Finally, an approach that implements controls without considering their impact on workflow or sample integrity may be ineffective or create new risks, demonstrating a lack of comprehensive risk management. The professional decision-making process for similar situations should involve a structured risk assessment methodology. This begins with hazard identification, followed by an evaluation of the likelihood and severity of potential harm. Control measures should then be selected based on the hierarchy of controls: elimination, substitution, engineering controls, administrative controls, and personal protective equipment. The effectiveness of these controls must be regularly reviewed and updated. This systematic approach ensures that all potential risks are considered and addressed in a manner that is both compliant with regulations and protective of laboratory personnel and the public. Question 17. What factors determine the most appropriate and comprehensive laboratory safety protocols and regulations that a clinical laboratory specialist in molecular biology, CLSp, MB, must adhere to in a U.S.-based laboratory setting? Option A. The interplay of federal regulations such as CLIA and OSHA, along with any applicable accreditation standards and professional guidelines, prioritizing the most stringent requirements. Option B. The minimum safety requirements outlined by the primary federal regulatory body overseeing laboratory operations. Option C. The safety protocols that are most cost-effective and easiest to implement for the laboratory staff. Option D. The internal safety policies developed by the laboratory's management, provided they are documented. The correct answer is A. The interplay of federal regulations such as CLIA and OSHA, along with any applicable accreditation standards and professional guidelines, prioritizing the most stringent requirements. Explanation: scenario analysis. This scenario is professionally challenging because it requires a CLSp, MB to navigate the complex and often overlapping requirements of multiple regulatory bodies governing laboratory safety. The challenge lies in identifying the most stringent and applicable standards when different regulations might offer varying levels of protection or focus on different aspects of safety. A failure to correctly interpret and apply these standards can lead to significant risks for personnel, patients, and the laboratory itself, including potential legal repercussions and loss of accreditation. Careful judgment is required to prioritize and integrate these requirements effectively. Correct approach analysis. The correct approach represents best professional practice because it prioritizes compliance with the most stringent applicable regulations and guidelines. In the U.S. context for a CLSp, MB, this means adhering to standards set by the Clinical Laboratory Improvement Amendments (CLIA) for laboratory quality and safety, the Occupational Safety and Health Administration (OSHA) for worker safety, and specific guidelines from professional organizations like the College of American Pathologists (CAP), if the laboratory is accredited by them. CLIA mandates a safe working environment and proper procedures. OSHA provides detailed regulations on hazard communication, personal protective equipment, PPE, and biosafety, and CAP, as a CLIA-approved accrediting body, often enforces even more rigorous standards. Integrating these ensures a comprehensive safety program that addresses all potential risks. Incorrect approaches analysis. One incorrect approach is to solely rely on the minimum requirements of a single regulatory body, such as only adhering to CLIA. This is professionally unacceptable because CLIA safety provisions, while essential, may not encompass the detailed occupational health and safety measures mandated by OSHA, particularly concerning chemical hygiene, bloodborne pathogens, and hazard communication. Another incorrect approach is to prioritize internal laboratory policies over external regulations. While internal policies are important, they must align with and often exceed regulatory minimums. They cannot supersede them. Failure to do so risks noncompliance and inadequate safety protections. A third incorrect approach is to only implement safety protocols that are easily achievable or cost-effective. This is ethically and regulatorily flawed as it compromises the well-being of laboratory personnel and the integrity of patient care by neglecting necessary safety measures due to convenience or cost, directly violating the spirit and letter of regulations designed to prevent harm. Professional reasoning. Professionals in molecular biology laboratories must adopt a hierarchical approach to safety compliance. First, identify all applicable federal and state regulations, e.g., CLIA OSHA. Second, determine if any accrediting bodies, e.g., are involved, as their standards often build upon or exceed federal requirements. Third, review relevant professional guidelines and best practices from organizations like the American Society for Clinical Laboratory Science (ASCLS) or the American Society for Microbiology (ASM). The CLSp, MB, should then develop and implement safety protocols that meet or exceed the most stringent requirements across all these sources. Regular training, risk assessments, and updates to protocols based on evolving regulations and scientific understanding are crucial components of this decision-making process. Question 18. When evaluating the suitability of bioinformatics tools and databases for the initial genomic characterization of a novel viral pathogen identified in a clinical setting, which of the following approaches best aligns with the principles of accurate molecular diagnostics and regulatory compliance for a CLSp, MB? Option A. Utilizing the most recently released bioinformatics software, regardless of its validation status, to ensure the use of cutting-edge analytical capabilities. Option B. Prioritizing bioinformatics resources that are freely available and require minimal computational resources, even if their performance characteristics are not well documented. Option C. Relying on informal recommendations from online forums and social media groups for tool selection, as these often reflect current community consensus. Option D. Selecting tools and databases that are widely cited in recent peer-reviewed publications related to viral genomics and have publicly available documentation detailing their algorithms and validation studies. The correct answer is D. Selecting tools and databases that are widely cited in recent peer-reviewed publications related to viral genomics and have publicly available documentation detailing their algorithms and validation studies. Explanation. Scenario analysis. This scenario is professionally challenging because the CLSp, MB, must select appropriate bioinformatics tools and databases for a novel pathogen's genomic analysis, balancing the need for speed in a potential public health crisis with the imperative of data integrity and regulatory compliance. The rapid evolution of bioinformatics resources means that outdated or inappropriate tools can lead to inaccurate conclusions, impacting diagnostic development, epidemiological tracking, and therapeutic strategies. The CLSp, MB, must navigate a landscape of diverse tools and databases, each with varying levels of validation, scope, and suitability for specific molecular biology applications, all while adhering to the stringent regulatory framework governing clinical laboratory practice. Correct approach analysis. The correct approach involves prioritizing tools and databases that are well documented, validated for clinical or research applications relevant to infectious disease genomics, and have a clear lineage of development and maintenance. This aligns with the CLSp, MB, certification's emphasis on competency in molecular biology techniques and their application, which implicitly includes the responsible use of associated analytical tools. Regulatory frameworks for clinical laboratories, such as those overseen by the College of American Pathologists (CAP), or the Clinical Laboratory Improvement Amendments (CLIA) in the U.S., mandate that laboratory procedures, including data analysis, must be performed using validated methods to ensure accuracy and reliability. Utilizing resources with established peer-reviewed literature support, clear version control, and documented performance characteristics demonstrates due diligence and adherence to quality management principles, thereby ensuring that any conclusions drawn are scientifically sound and defensible within a regulated environment. Incorrect approaches analysis. Choosing tools or databases based solely on their popularity or ease of use, without regard for their validation status or suitability for the specific genomic data type. E.g., whole genome sequencing, targeted sequencing, represents a significant regulatory and ethical failure. This can lead to the generation of erroneous results, violating the CLIA requirement for accurate and reliable testing. Relying on unverified or experimental bioinformatics pipelines without proper validation also bypasses the established quality control measures expected in a clinical laboratory setting, potentially compromising patient care and public health. Furthermore, using databases that are not regularly updated or curated for the specific pathogen or gene families of interest can lead to missed critical variants or misinterpretations of sequence data, directly impacting diagnostic accuracy and research integrity. This failure to employ appropriate, validated methodologies is a breach of professional responsibility and regulatory compliance. Professional reasoning. When faced with selecting bioinformatics tools and databases for a novel pathogen, a CLSp, MB should employ a systematic decision-making process. This involves 1. Clearly defining the analytical objective, e.g., variant calling, phylogenetic analysis, gene function prediction. 2. Identifying potential tools and databases relevant to the objective and pathogen type. 3. critically evaluating the documentation, validation status, and peer-reviewed literature supporting each resource. 4. Assessing the suitability of the tool/database for the specific data type and experimental context. 5. Considering the maintenance and update frequency of the resource. 6. Prioritizing resources that have demonstrated performance in similar clinical or research applications and are supported by robust quality management systems. This structured approach ensures that the chosen bioinformatics resources are appropriate, reliable, and compliant with regulatory expectations for accurate and reproducible molecular diagnostics and research. Question 19. Cost-benefit analysis shows that a novel, open-source sequence alignment algorithm offers significantly faster processing times compared to established, commercially validated tools. However, this new algorithm has limited peer-reviewed validation data, specifically for clinical diagnostic interpretation of complex genetic variants, which approach best aligns with regulatory requirements and professional best practices for a CLSp, MB, in a US-based clinical laboratory. Option A. Utilize the novel algorithm for initial screening, and then realign critical findings with the established algorithm to mitigate risks, without formally validating the novel algorithm's performance in this dual approach. Option B. Develop a custom alignment algorithm in-house, leveraging the principles of the novel algorithm but without formal validation, to achieve a balance between speed and cost. Option C. Continue using the established, commercially validated sequence alignment algorithm, prioritizing its documented accuracy, reliability, and regulatory compliance for clinical diagnostic reporting, even if it incurs higher costs and longer processing times. Option D. Implement the novel algorithm due to its speed and cost-effectiveness, assuming its general alignment principles are sound and will suffice for clinical interpretation. The correct answer is D. Implement the novel algorithm due to its speed and cost-effectiveness, assuming its general alignment principles are sound and will suffice for clinical interpretation. Explanation. This scenario presents a common challenge in molecular diagnostics, selecting the most appropriate bioinformatics tool for a specific clinical application while adhering to regulatory standards. The CLSp, MB, must balance the need for accurate and reliable results with the practical considerations of resource allocation and efficiency. The professional challenge lies in interpreting the nuances of different sequence alignment algorithms and their suitability for clinical decision-making, ensuring that the chosen method meets the stringent requirements of diagnostic accuracy and regulatory compliance. The correct approach represents best professional practice because it prioritizes the validation and regulatory compliance of bioinformatics tools used in clinical diagnostics. In the U.S., the CLIA Clinical Laboratory Improvement Amendments regulations mandate that all laboratory testing, including molecular diagnostics, must be performed using methods that are accurate, reliable, and have been adequately validated. The CLSp, MB must ensure that any sequence alignment algorithm used for clinical interpretation has undergone rigorous analytical and clinical validation, demonstrating its performance characteristics, sensitivity, specificity, accuracy for the intended use. This aligns with the FDA's oversight of laboratory-developed tests, LDT, and in vitro diagnostic IV devices, which requires appropriate validation and quality control. Choosing a tool that has established performance metrics and is supported by peer-reviewed literature or regulatory submissions is crucial for patient safety and reliable diagnostic reporting. An incorrect approach that relies solely on the speed or perceived ease of use of a bioinformatics tool without considering its validation status for clinical applications is professionally unacceptable. This failure directly contravenes CLIA regulations, which require validated methods. Using a tool that has not been analytically validated for the specific clinical context could lead to misinterpretation of sequence data, resulting in incorrect diagnoses, inappropriate treatment decisions, and potential patient harm. This also represents a failure to adhere to good laboratory practices, GLP, and potentially the FDA's requirements for diagnostic devices, depending on the nature of the test. Another incorrect approach would be to select a tool based on its popularity in academic research without assessing its suitability for the clinical workflow and its documented performance in a diagnostic setting. While academic tools may be cutting edge, they may lack the robustness, documentation, and quality control necessary for routine clinical use. This oversight can lead to inconsistent results and a lack of confidence in the diagnostic output, violating the principle of providing reliable patient care. The professional decision-making process should involve a systematic evaluation of available sequence alignment algorithms. This includes 1. Defining the specific clinical question and the type of sequence data being analyzed. 2. Researching algorithms with documented performance characteristics for similar applications. 3. Assessing the validation status of the algorithm for clinical diagnostic use, including any regulatory clearances or approvals if applicable. 4. Considering the algorithm's integration into the laboratory's existing workflow and IT infrastructure. And 5. Ensuring that the chosen algorithm is supported by appropriate quality control measures and ongoing performance monitoring, all within the framework of CLIA and other relevant U.S. regulatory guidelines. Question 20. Strategic planning requires a CLSp, MB, to develop a new molecular assay for detecting a specific pathogen. A critical step in this assay involves visualizing amplified nucleic acid fragments via gel electrophoresis. The CLSp, MB, must determine the optimal agarose gel concentration to achieve clear separation of fragments within a defined size range. Which approach best addresses this implementation challenge while adhering to regulatory expectations for clinical molecular diagnostics? Option A. Choose the lowest possible agarose concentration to maximize fragment migration speed, thereby reducing assay turnaround time without assessing its impact on resolution. Option B. Immediately adopt the agarose concentration used in a published research paper for a similar assay, assuming it will be directly applicable without further testing. Option C. Select a high agarose concentration, e.g., 0%, to ensure maximum resolution, even if it means significantly slower migration and potential difficulty in resolving larger fragments. Option D. Systematically test a range of agarose concentrations, e.g., 0, 8% 1, 0% 1, 5% 2, 0%, and evaluate the resolution, band sharpness, and migration patterns of the amplified fragments for each concentration, selecting the concentration that provides the clearest and most distinct separation within the expected size range. The correct answer is D. Systematically test a range of agarose concentrations, e.g., 0, 8% 1, 0% 1, 5% 2, 0%, and evaluate the resolution, band sharpness, and migration patterns of the amplified fragments for each concentration, selecting the concentration that provides the clearest and most distinct separation within the expected size range. Explanation. Scenario analysis. This scenario presents a common implementation challenge in molecular diagnostics, optimizing gel electrophoresis for a novel assay. The challenge lies in balancing the need for robust, reproducible results with the practical constraints of time, cost, and reagent availability. A CLSp, MB, must not only understand the technical principles of gel electrophoresis, but also how to adapt them to specific applications while adhering to regulatory standards for laboratory practice. The pressure to deliver results quickly for clinical decision-making adds another layer of complexity, requiring a systematic and evidence-based approach to method development and validation. Correct approach analysis. The correct approach involves a systematic evaluation of gel matrix properties, specifically agarose concentration, to achieve optimal separation of target nucleic acid fragments. This aligns with best professional practice by prioritizing empirical data and established principles of molecular biology. Regulatory frameworks for CLSp, MB, emphasize the importance of method validation and ensuring that laboratory procedures are fit for purpose. By testing a range of agarose concentrations, the CLSp, MB, is gathering the necessary data to select the concentration that provides the best resolution and sensitivity for the specific amplicon size range, thereby ensuring the accuracy and reliability of the diagnostic test. This data-driven approach is fundamental to maintaining quality control and meeting the standards expected in a regulated clinical laboratory environment. Incorrect approaches analysis. One incorrect approach involves solely relying on a previously published protocol without independent verification. This fails to account for potential variations in reagents, equipment, or sample matrices that can significantly impact gel electrophoresis performance. Ethically and regulatorily, This is unacceptable as it bypasses the crucial step of method validation, potentially leading to inaccurate results and compromising patient care. It demonstrates a lack of due diligence and adherence to quality assurance principles. Another incorrect approach is to arbitrarily select a high agarose concentration without considering the amplicon size. While higher concentrations can improve resolution for small fragments, they can also impede the migration of larger fragments, leading to poor separation or complete failure to resolve. This approach lacks a scientific basis and ignores the fundamental relationship between gel pore size and nucleic acid fragment size. This can lead to false negatives or ambiguous results, which is a direct violation of the CLSp MBS responsibility to ensure test accuracy and reliability. A third incorrect approach is to prioritize speed over resolution by using a low agarose concentration without assessing its impact on the assay's sensitivity and specificity. While faster migration might seem appealing, it could result in smeared bands or poor separation, making it difficult to interpret results accurately. This compromises the diagnostic integrity of the assay and fails to meet the expected standards of performance for a clinical laboratory test. It represents a failure to adequately validate the method for its intended clinical use. Professional reasoning. Professionals in this role should adopt a systematic, data-driven approach to method development. This involves 1. Clearly defining the performance requirements of the assay, e.g., expected amplicon size, required resolution. 2. Consulting relevant literature and established protocols as a starting point. 3. Designing experiments to empirically test critical parameters, such as agarose concentration, buffer conditions, and running voltage/time. 4. Analyzing the results of these experiments to identify the optimal conditions. And 5. Documenting all experimental procedures, results, and decisions for future reference and regulatory compliance. This iterative process ensures that the developed method is robust, reproducible, and suitable for clinical application.